So we're gonna finally start getting into living things today. Although we will do one last little nod to chemistry because there's one last little thing we need to add in. And I've mentioned before, but I'll mention again, the quiz will be after lecture, before we start lab. So we'll do lecture. There's no warm up today because it seems silly to do a warm up if you got a quiz anyway. So we'll do lecture, we'll take a break, we'll do the quiz, we'll take a break, and then we'll get into lab. Okie dokie. So when we were talking about all the different, what are the basic kinds of biomolecules we were talking about? Main classes.
Proteins,
lipids,
carbohydrates,
Proteins, lipids,
nucleic acids,
nucleic acids.
carbohydrates, nucleic acids. So there are also a lot of important molecules that are hybrids, that have parts of different things that are mixes of proteins and carbohydrates, and or mixes of lipids and carbohydrates, or lipids and proteins and this and that. So I'll just give you some vocabulary so you kind of have a sense of what kind of cast of characters we'll be meeting as the semester goes on. So important ones, if you have things that are mainly protein, but with some carbohydrate, with some sugar groups on them, then those are glycoproteins. Glycoproteins are mainly protein, with carbohydrate groups, sugar groups hanging off of them. These are going to be really important in the cell membrane. We're going to see a little later. Hold on, I'm going to mute Natalia here. So in fact, a big part of the way your cell has what it looks like from the outside is all these glycoproteins embedded in the membrane, giving it specific looks. So glycoproteins are just proteins with sugar groups, again, glyco sugar. There are also molecules that are also a mix of carbohydrate and protein, except if they're mainly carbohydrate with like amino acid groups hanging off, then it might be a proteoglycan. So a glycoprotein and a proteoglycan, they're both hybrids between protein and carbohydrate, but this one is mainly a protein with some sugar groups. This is more mainly a sugar with or a carbohydrate with some amino acid peptide groups. Again, you're making glycoproteins, we'll talk about more, but it's, after you build the protein, there's more machinery. Remember all the Golgi bodies and stuff in the cells. So you can take things and you move them from one compartment to another, and you have all the right enzymes to do all the modifications to add in the different groups and stuff to make the actual molecule. Other things we'll see, if you have carbohydrates added to lipids, you can get glycolipids. These are things that are mainly lipid, but then have sugar groups added on. These are also things you can find in membranes and in other places. Another really important hybrid molecule that we're going to see that's important for transporting fats and lipids around in the bloodstream are the 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 lipoproteins. So lipoproteins, mixes of lipid and protein. You know, the way we transport lipids around, and fats don't dissolve in water, as you all well know at this point. But if you take a fat, some lipid, and you add some protein around it, this would be like a lipoprotein. You know, it's got some protein on the outside, the lipid kind of in the middle. This thing will dissolve in your plasma, and this is how you transport, you know, fats around. We're going to talk about the HDLs, the high density lipoproteins, the LDLs, the low density lipoproteins, for moving fats around from one place to another, either depositing them or getting rid of them. So just kind of putting it out there that we've reviewed all these basic molecule types, but then there's lots of these hybrid molecules that are going to be important as well. All right. So now we can get into finally this basic kind of structural and functional unit of life, right? The cell. You know, we're gonna be spending a lot of time talking about some of the basic properties of cells. In fact, from now until we get to the first exam, we are basically just talking about cells, because you're made up of tens of billions of cells that all connect up together and coordinate. So we have to kind of understand the basic cell before we start putting them together to make the more complicated things, your tissues and organs and things like that. Does anybody remember like why they're even called cells?
Because they contain a nucleus.
No. No, in fact, there's a lot of cells that don't contain a nucleus, like all the prokaryotic cells don't have a nucleus even. I mean, that word, what does it sound like?
Sounds like a little
Cellular?
room.
Sounds like a little room, right? It's like if you do something wrong in the eyes of the state, you're going to end up incarcerated in some little cell. And when was Hooke, Robert Hooke, I think, back like hundreds of years ago, when he had his first kind of primitive microscopes, looked at little slices of cork under the microscope. And you know, it was actually the cell walls left over from the plant cells, but you know, it looked kind of like this. Plant cells tend to be more rectangular, so it looked to him like little rooms. For him, it wasn't the cells he'd lock somebody in a jail. It was more like the little cells that a monk would be in, kind of transcribing some book or something. But it was supposed to just be a little room, so it gets its name from little room. You know, the reality is the cells we're going to see in this class are going to range from things that really do look kind of just like simple little like classic looking cell. We're going to see cells like neurons that have literally 100,000 interconnections to other things with these crazy little processes coming over. We're going to see skeletal muscle cells, which are these huge things that are literally like over a foot long and have hundreds of nuclei. So, you know, what a cell actually is a huge range of possibilities. So they come in lots of different varieties, but we're going to kind of look at some of just kind of the basic stuff about cells right now. But remember that actual cells are really varied. So let us talk about basic parts and pieces, which you should all be pretty familiar with from your bio, your general bio, introductory bio. So what are some of the classic organelles of a cell?
Mitochondria.
Mitochondria. You know, what's happening in mitochondria?
Making ATP.
Yeah, that's going to be where the majority of your ATP is getting made. We're going to be spending a lot more time in the mitochondria in the coming week, looking at the details and all the metabolic pathways going on in there to extract all the energy from glucose and make lots of ATP for the cell. What are other important organelles?
Cell wall.
A cell wall only exists in a plant. A human cell, animal cells don't have cell walls. Cell walls are made out of cellulose, and they help stabilize the thing, especially with kind of internal water pressure.
The smooth endoplasmic reticulum and the rough endoplasmic reticulum.
Yeah, so there's that, the endoplasmic reticulum, which are basically these passageways of membrane inside the cell. Endoplasmic reticulum can play different roles. She was saying rough. What makes rough endoplasmic reticulum rough?
Ribosomes.
Ribosomes. They got ribosomes added on there. You can also have free ribosomes as well. What do ribosomes do?
Is it something with protein synthesis, if I'm remembering that correctly?
Yeah, exactly. They're the site of protein synthesis. That's where the messenger RNA gets brought to, and then the transfer RNA will bring the appropriate amino acids and assemble them in that primary structure to make the proteins. That happens at ribosomes. In rough ER, since the ribosomes are on the ER itself, you can start assembling the amino acids and just thread them directly into the endoplasmic reticulum. So they're now in this, passageway made out of membranes, and they can go get moved to where they need to get moved or be adulterated by different, maybe they have to add some sugar groups to them, who knows. Smooth ER doesn't have ribosomes. It tends to have more enzymes actually attached into the membrane. So it's a place more for like processing stuff versus assembling and storing proteins and things. You find a lot more smooth ER, like, in liver cells or cells that are doing a lot of kind of processing of things. What other organelles are important?
Golgi apparatus.
The Golgi apparatus. It's another membranous organelle. But instead of being kind of more these big interconnected pathways, the classic picture is like kind of a stack of pancakes or something. We're just talking about, like glycoproteins. A glycoprotein, obviously, if it's a protein with sugar groups, it has to start out as a protein. You make a protein at the ribosomes, but then once you have that protein, we need to take it and we can move it from, it's basically like an assembly line. You can take this protein and you put it into these other vessels where different chemical reactions happen that add on the sugar groups and create the final molecule. And then the Golgi then also can package and move things around. So the Golgi apparatus is a way to, kind of do this post-processing of things, for instance. What are other important?
Cell membrane.
Say it again.
Oh, cell membrane.
Cell membrane. Yeah, the cell membrane is going to be hugely important in our class. The cell membrane has a few synonyms. Let me get this looking better. Cell membrane, also called the plasma membrane, also called the plasma lemma. In fact, once we kind of are done with this kind of list of organelles, we're going to spend the rest of today just getting more into detail about the cell membrane and its properties and different kinds of proteins embedded in the cell membrane and all the different kinds of jobs they do. So the cell membrane is going to be hugely important in our class because it's that boundary between you and the rest of the universe, right? This is where you either have stuff inside the cell and then everything outside the cell. It's also how your cells are going to have ways to coordinate with all the other cells. That's going to be like kind of the eyes and ears are going to be on the cell membrane. So we're going to talk a lot about the cell membrane. Other important parts of the cell.
The nucleus.
The nucleus, totally. So the nucleus is kind of typically in the middle, but not always. Nucleus has a double membrane around it, actually, as do like the mitochondria. What's the main thing we think about the nucleus? What's the nucleus doing?
Holding the DNA.
It's kind of storing the DNA. It's like where you have kind of the blueprint of how to make all the stuff the cell needs to make. It's also where you have a lot of the regulatory machinery, like turning things on and off. How does the cell know it's time to divide? Or how do I know I need to make this or not make this? So, you know, the DNA in there partly is blueprints on how to make stuff, but there's also a huge amount of just regulation of when to do things and how to do things and what to turn on or turn off and all that. Right, you know, if some of that regulation breaks, doesn't work right, that's one of the ways you get cancer. The cell starts thinking I need to divide or I need to make extra things when it shouldn't be. So nucleus, you know, I'm assuming you all have basic understandings of DNA and DNA getting transcribed to messenger RNA and messenger RNA going off to the ribosomes and ribosomes assembling into proteins. But, you know, beyond that for this class, we're not going to get much deeper in that. Kind of what you learned in your Bio 110 is going to serve you fine. What are other important organelles?
Cytoskeleton.
So cytoskeleton, totally. So, you know, this is not just like a Ziploc filled with goo and stuff. You know, most cells have a very distinct shape that they hold. Most of the organelles are held in place on the scaffolding that keep them where they are. A lot of the stuff that gets moved around from one place to another is actually like a railway system. There's all sorts of specialized little tubules that things get pulled and dragged along. So let me, here's my cell again. Let's talk about some cytoskeletal elements. There are microfilaments. Microfilaments are basically actin. We'll meet actin in more detail later when we're looking at myosin actin and the cross bridges and muscle contraction. But actin is also just a really important cytoskeletal element. It's like this little molecule that assembles into these, what we call thin filaments. And those are like what are holding the nucleus in place. That's why the nucleus stays in the middle. There's actin holding it there. We're going to talk about like microvilli, things to like increase the the surface area of cells. Like if you took just a piece of cloth and you pleated it into little pleats and you let go, they would all just undo. You know, if you want to have little folds for microvilli and you want them to stay, you need to have some kind of cytoskeletal structure to hold that. That's going to be actin, little microfilaments that hold the little microvilli, the little folds to increase the surface area. It's also going to be important in motility and movement, and we'll see very dramatically, particularly in like muscle contraction. So microfilaments, microtubules are going to be super important. Microtubules are made out of these little tubulin subunits. And you can assemble them and you can grow the microtubules. You can kind of break them apart and you can shrink the microtubules. Right, these are what make up the spindle in mitosis. The spindle grows, its little microtubules growing out, grabbing onto the sister chromatids and pulling them apart as they shorten and all that. Microtubules also are going to be important kind of as a railway system in your cells when we're going to have specialized little proteins. Like we're going to talk about like a neuron. You know, some neuron is literally could be like 3 feet long. There's a neuron that goes from the base of your spinal cord to the tip of your toe. That means you have to get stuff from one side of the cell to the other side of the cell. You know, there's going to be microtubules and there's going to be specialized little motor proteins that drag things back and forth along the microtubules, kind of like little like railway cars or something. So microtubules play a role for that. Microtubules are going to be at the core of how cilia move back and forth and wiggle, and or how the flagellum on a sperm does its thing so a sperm can swim towards its destiny with the egg. So we'll be seeing microtubules. Microtubules tend to have, I'm saying that they can get built up. There's things called centrioles, or like these microtubule organizing centers. They're the places where the microtubules are kind of getting built up or shrunken down. Sometimes there's also things called basal bodies, especially in our cilia. We'll see there's little things, little areas where the microtubules are kind of anchored and grow from. So microtubules are going to be important. And then there's a whole slew of other things. There's kind of a catch-all term we call intermediate filaments. This is kind of a catch-all. Example is like keratin, for example. What, not keratin. This is the fiber in your skin cells that kind of makes your skin kind of tough. You know, so you scratch your, your skin obviously is the external skin. I compare the skin inside your mouth, which does not have the keratin as much as compared to the skin outside of your body that has that kind of more tough scaliness. So, there's lots of different filaments that are going to be important that are not actin or microtubules. And so they just get lumped into this. So we'll be spending time looking at cytoskeleton. Cytoskeleton is going to be important for motility, right? We're going to, in fact, a little bit, we'll talk about, endocytosis, exocytosis. The classic thing of, oh, here's a cell and here's something outside the cell. And then the cell like reaches around and it pinches it off. And now this thing is inside the cell, right? This would be like endocytosis, the classic thing. It's like cells, cell membrane is just phospholipids. It doesn't move on its own. The only reason this can happen like this does is there's all sorts of cytoskeletal things pushing and pulling and, deforming this cell and making it do this. So this is a very active, in fact, this is a version of active transport we'll be talking about, endocytosis, exocytosis. And it's only going to be possible if there is all sorts of cytoskeletal elements pushing and pulling on the membrane to make the cell do this thing. Cells don't just spontaneously become a Pac-Man or something like this. So cytoskeleton will be important for lots of things. Okay, dokie. So cytoskeleton, that's a good one. What's another important cellular organelle?
Lysosomes.
Lysosomes. Lysosomes are little vesicles that are filled with digestive enzymes to help kind of break things apart and recycle them. Either sometimes cells eat things and they need to break them down, or cells are constantly, recycling and repairing their own stuff inside. So they're going to constantly have to be breaking things down. So you can take a vesicle that's filled with whatever, a vesicle that is a lysosome filled with digestive enzymes, and you fuse them. And then those digestive enzymes can help break down whatever you want to break down. It's part of the reason you don't ever want cells to just die and just spill out their guts, because that's going to cause a lot of damage to all the surrounding cells, right? If this cell just died and just kind of broke apart and just kind of spewed out into the surrounding back. It's like all of the digestive enzymes in these lysosomes would also be just spewing out, and all of a sudden we'd just be damaging everything around. So when one of your cells dies, it's much more of a controlled thing. Like one of the words we'll be talking about, apoptosis. This is controlled cell death. So like when a cell is going to die, it tends to do it in this very intentional way and kind of fragment itself into little bits of contained stuff that can then get cleaned up by the other cells of like your immune system and stuff. So when a cell dies, it doesn't just fall apart, because again, that would be very damaging to everything around it. So they tend to die in this specific program that allows them to die and get cleaned up without causing peripheral damage or collateral damage to the surrounding tissues. So yeah, well, this word will come back. So I think we've kind of gotten into a lot of, most of the most important cellular organelles. You should, I'd feel perfectly fair to put something on your next exam asking like, what's the job of a ribosome or the mitochondria or this or that. You don't have, we're going to go much more into detail about certain ones of these, but you should know the basic functions of the basic organelles of a cell. But what we're going to do now is we are going to be focusing on the cell membrane. So that's what we're going to be spending the rest of today doing. And it's going to be a focus for a lot of the things for the whole rest of the semester even. So we need to spend some more time talking about cell membrane. So again, what are the main kinds of molecules that make up the cell membrane?
The phospholipids.
The phospholipids, exactly. So remember, if you remember, hopefully you all remember, phospholipids had that hydrophilic head and the hydrophobic tails, and they spontaneously start assembling into this phospholipid bilayer. Again, the heads are polar, so they will want to be out towards the water. The tails are nonpolar and therefore kind of make this hydrophobic region in the middle here. Then we also talked about cholesterol intercalating in between. Again, the more cholesterol you got, the more kind of waterproof the membrane is. The more cholesterol is, the less things can kind of just shift around, kind of loosey-goosey, kind of packs things in. Because again, the only thing holding the phospholipids together here are the hydrophobic hydrophilic interaction. The reality is everything is kind of shifting. We're going to talk right now in much more detail about what's called the fluid mosaic model of the membrane. The idea of fluid mosaic model, fluid meaning that the phospholipids and other parts are not like, glued in any permanent way, that they're fluid, they can shift. The tails are always going to stay facing inward, the heads are always going to face outward. But if you looked at, focused on any one particular one at a moment, it's kind of shifting and, moving and kind of on its just kind of random journey floating around. Fluid mosaic model, mosaic meaning that there's lots of different things in addition to the phospholipids, primarily the membrane proteins. So let me put that in here. So the membrane proteins, and the ones I've drawn here are membrane, they're spanning. Some of the membrane proteins are only like on one side. They don't necessarily go all the way across. Some of them just stay on one side. But they're also part of this fluid mosaic model, meaning they're kind of moving around. What's going to keep a membrane protein in place in the membrane like this?
Outside? Like, wouldn't it be like the outside of the layer, right? Like that's holding it, because
No, it's going to be the same things that the phospholipids together, actually.
The cholesterol or
No,
Part of the cytoskeleton?
no,
Interactions with the hydrophilic and hydrophobic regions.
exactly. It's going to be just the same thing, the hydrophobic, hydrophilic. If you kind of go back and remember, we talked about some amino acids have the side chains that are hydrophobic or nonpolar. Some have side chains that are hydrophilic, or like make hydrogen bonds and all that. So what happens is you end up with this protein and you have, amino acids that make these two ends are polar. And you have this middle part that's nonpolar. And so it's going to want to just stay exactly situated like this. If you try to push this up this way, this polar part is going to want to pull back down because it wants to be near there. It doesn't like to be in this nonpolar region, and this nonpolar region tends to situate itself in this nonpolar region here amongst all the nonpolar tails. You know, if I try to pull this the other way, the same thing. It's kind of whichever way you push it, it wants to just be right like this, with the two polar regions facing outside and inside, and this nonpolar region being situated in the nonpolar region of the membrane. So this, again, just like the phospholipids, is fluid. It can actually migrate and move around. It doesn't have to necessarily be next to, you know, it might say like, hi there, Mr. Phospholipid, nice to meet you, but then it drifts away and then it never sees this one again because now it's drifted over to some other part of the membrane because things aren't glued together in any particular way. It's just about these hydrophobic, hydrophilic interactions that's holding everything together. Does that make sense? Let me, hey, wait a second. I'm going to, oh, this, what's going on? I actually have my PowerPoint thing here, but it's being weird. So give me a second here. Okay, that's working. All right. Here we go. I'm going to share. Right, so this is just from your book, you know, kind of showing what the cell membrane looks like with a little bit of a better drawing than I could do. You know, each of these little purple things here are the phospholipids with their heads and their tails. You can see the little cholesterol molecules kind of intercalated in between. And then you can see these proteins. Here's these transmembrane proteins. And again, everything's just kind of floating around with respect to each other. They hold their basic structure as a phospholipid bilayer. But again, fluid mosaic model, meaning fluid, things can shift with respect to each other. And mosaic, meaning there's lots of different things all making up the cell membrane. Yes, there's phospholipids as the primary thing, but then lots of proteins. And again, a lot of these proteins are glycoproteins, meaning they have these sugar groups attached to them. You know, some of these, this thing here is supposed to be a protein, a membrane protein showing a little more detail with these 7 transmembrane helices. Does anybody remember what do the helices result from? Why are there helixes in this protein?
The alpha.
Yeah, that's part of that secondary structure, the alpha helices. A lot of the proteins that we're going to see are going to have these transmembrane domains that go back and forth to create either different kinds of structures, receptors, or tunnels or things for here. So this is just supposed to be a more complicated version of what a protein might look like as it's folding up in these complicated ways, secondary, tertiary structure. So sometimes the proteins are anchored, like for certain cells, like in your epithelial cells, it's important that you have apical. The apical side of the cell has different kinds of proteins compared to the basal side. So sometimes you actually do anchor the proteins and keep them on one part of the membrane or another. Other times they're just kind of loose to migrate in the more fluid mosaic model. But just kind of getting in your head, this is what a cell membrane really is like. It's this assembly of all these little phospholipids with all these other membrane proteins in there. And we're going to spend a bunch of time today looking at the jobs, the roles that these different membrane proteins can play. Let's clear that.
Would those alpha helices sort of act like a railroad in transport?
Not really. We'll get back to see different roles they play.
Okay.
The distances that we're looking at across the membrane are so tiny. When I was talking about the railroad thing, I was talking about dragging things across, much longer distances from one part of the cell to the other, rather than like across the membrane of the cell. So the processes that move things across the membrane is going to be a little different. This is actually just worth looking at. Phospholipids can be in this kind of more bilayer, making the kind of main cell membranes. We're going to see phospholipids forming little micelles. This is going to be important. Like if you have a lipid that you want to absorb from your diet, you can have it at the core here, and then it gets kind of surrounded by all these phospholipids. This is going to basically be kind of how bile helps you absorb fats from your diet. Liposomes are kind of like a little vesicle. This is actually like how, like when you're trying to get drugs into cells, I think this is actually how the COVID vaccine works, is by kind of getting the RNA into little liposomes, which can then fuse with the cell membrane and then move into the cell itself. So having phospholipids kind of just surrounding some little droplet in here is a really useful way of trying to actually deliver drugs into a cell, because you can take this, and when this comes up to here, it can then fuse and dump out its contents, its cargo, into the cell. So blah, blah, blah. Let's see if there's more things in this. No, you can use this picture. Before I get into the so this version of this is kind of showing the cytoskeleton. I think it's kind of interesting. You can see microfilaments. You can see here these microvilli are being supported by little actin. Microtubules are helping drag things around, hold things together. Well, there's a chat. Okay, let's stop sharing this. All right, so what we're going to talk about now are all of these different membrane proteins that are in the membrane playing different roles. And we do need, a lot of them are going to be major players for the rest of the next 3.5 months. So we're going to spend some time going into the details of what they are, the different subtypes and all that. So let's do that. So roles of membrane proteins. And again, by membrane proteins, these are just proteins that are sitting there within the cell membrane and in this fluid mosaic model, sometimes spanning across the entire membrane, sometimes either on the inside or the outside of the cell, but kind of stuck in the membrane due to the hydrophobic, hydrophilic interactions that holds the lipid bilayer together. So, what are some of the roles of membrane proteins?
Transport.
So a big one is transport. And so I never know which one we're going to start with, because I like kind of let people kind of come up with things. So we'll start with transport. Transport's going to be a deep enough one that we're probably not going to even finish transport before we're done with our lecture today. So we'll be continuing with other membrane proteins as we continue the week. So transport. Before we get into more detail here, let's just remember a phospholipid bilayer, a cell membrane. So without any help, if there was no membrane proteins or anything, if I'm just thinking about the permeability of a cell membrane, what do we know about what can cross, what can't cross without any other things coming into the picture?
Non-polar can enter the cell.
So non-polar, yes. So things that are non-polar don't need any help at all. As long as they might need help getting up to the membrane because they can't just dissolve in water. So we're going to see all sorts of special carriers that are moving steroid hormones around from one place to another. But once they get to the membrane, they can drift across from one side to the other without help, because non-polar things go into the non-polar region of the tails. What else do we know about permeability of a cell membrane?
Water can get through.
So water, and what is it about water that allows it to do it?
It's tiny.
Yeah, so very tiny polar. Typically, though, if things are polar, they don't cross. They're going to stay pulled on one side or the other side with all the hydrogen bonding with the water, and they're not going to venture into that hydrophobic region between all those hydrophobic tails. So in general, polar, in general, no. So non-polar and very tiny polar things can get across without help typically. Polar things in general, not. And there's a lot of polar things that we really want to cross the membrane, like glucose. You know, if a cell's going to get energy, it needs to get absorbed some glucose. But glucose does not cross a membrane without help. We're going to have membrane proteins that bring glucose from one outside of the cell to the inside of the cell. We talked about ions because of that hydration sphere. That's, what do they call the solvation shell? Because all of the water molecules are so attracted to that strong, to the really strong charge of an ion actually acts kind of like a big snowball. So an ion is not going to be able to cross the membrane without specialized membrane proteins to help them move across. So we're going to talk about movement across the membrane. So let's talk about transport proteins. Things that are going to help typically polar molecules come across. So one are called channels. These are typically for ions. Typically, they are for specific ions. So we're going to talk about potassium channels or chloride channels or calcium channels or sodium channels. Sometimes they are more for classes. Sometimes you'll have things that are just cation channels that'll let in, you know, potassium or sodium, or sometimes we'll have divalent cation channels that'll let in magnesium or calcium. Sometimes we'll have just leak channels that will let in any kind of ion. But very often we'll have channels that are specific for a particular kind of ion. So, but again, they come in different flavors. I shouldn't say different types. But a lot of the more interesting ones we're going to see are going to be specifically letting in one kind of ion versus another. And they're like, they're like a tunnel. Once they open, things can move in or out. I should mention, let's just review diffusion for a second. What is diffusion?
Higher concentration to lower concentration.
Yeah, so, some solute moves from higher to lower concentration. Right, so if I have some cell with some, let's say some sodium channel here. If I'm just thinking about diffusion, this thing is just going to move in or out depending on its concentration gradient. If I have a whole lot of sodium out here and not much in here, the sodium will move in. If I had a whole bunch of sodium inside and not much out, the sodium would move out. They're not like moving it in any particular direction. They're just making like a tunnel for it to move based on diffusion, or as we're going to see later with ions, based on electrical gradients as well. So we call this passive transport. Things are going to move based on like diffusion gradients and stuff, or concentration gradients. Sometimes they're just open, but the ones that are more interesting to us are gated. So these can be gated. So the way to think about this is think about a doorway. You could have a doorway without a door on it, and then people can just come in and out, and you can't control whether that doorway is open or closed. But you're gonna finally get to here. You get to finally meet. This is Nimbus. Hey guys. He's like, I'm not interested in these cells. All right, hold on. Yeah, usually he's more social than this. All right, I see John's got a black cat. Yeah, make sure you keep him in on Halloween so he doesn't get in trouble. All right, so gated channels, it's like a door. You have the door shut to your house, and as long as the door is shut, people can't come in or out. And if you want people to come in or out, you can open up the door, and then they can come in and out. And then later on, you can close the door and they can't come in and out anymore. That's what we mean by gated channels. They're channels that if they're open, the ions can move in and out, like I just showed in that previous moment. And if they're closed, then it's as if there's no channel at all. So what we want to talk about, what kind of things, what can control a gated channel? What can determine whether that channel is open or closed at any particular moment?
Charges?
Say it again, Catlin?
Charges?
Yeah, so charges. So it can be, can be what we call voltage gated. Let me explain that a little more because it's going to be an important concept for our class. So I've mentioned voltage as being a separation of charge, right? If you have a cell and there's just, let's say there's just an equal balance of negative ions inside and positive ions inside and outside. There's no net imbalance of charge. If I take some cation here, I could take this thing. It doesn't care which side it's on. If it's out, it doesn't necessarily want to go in because it's not more negative or positive inside or outside. So if I have no charge imbalance, then there is no voltage. The voltage is this kind of electric potential. But if I do create a charge imbalance, let's say I decide I'm going to take a bunch of the positive charges that are inside my cell here, and I'm going to move them out of the cell. So I am going to create this charge imbalance now where there is more positive outside and more negative on the inside. This is now a situation where we say there is a voltage across the membrane. So I'm going to draw it. Let's see. Usually we kind of draw it like this. V sub M, the membrane voltage. By the convention that we use, if the membrane voltage is negative, it means the inside is more negative with respect to the outside. If the membrane voltage is positive, we mean that there's more positive inside compared to the outside. So it's always inside with respect to outside of cell. So in this case that I've drawn here, we'd say the cell has a negative membrane voltage because it's more negative on the inside compared to the outside. What this means now, if I take my cation here, if it had its druthers, would it rather go into the cell or out of the cell?
Into.
Oh.
It wants to go into the cell because opposites attract. The positive ion wants to go in towards more negative. So this membrane voltage creates a electrical field that is going to actually create a force, drive the motion of ions, which is going to be really important as we continue on. So we're going to see, particularly in excitable cells like neurons and muscle cells, most cells tend to have a resting membrane voltage of around -70. So I'll write that just as a little teaser for a little later. -70 millivolts. Volts is just the measure of electric potential. And there are channels that will open or close based on what this membrane voltage is. So like a lot of the ones that we're going to see next week, looking at when we look at how neurons create their action potentials and things, they're going to be closed when the membrane voltage is -70 millivolts, but they'll open when the membrane voltage goes up to -55 millivolts. So when I'm saying that a ion channel can be voltage gated, it means that it's going to monitor the voltage across the membrane, and in one condition it might be closed, in another condition it's going to be open. Again, this is going to be at the core of the electrical signaling in your neurons, is these ion channels that are going to normally be closed, but as things start getting more electrically excited, they'll open up and start creating these chain reactions, the action potentials that are going to run from one side of the cell to the other. It's also going to be at the core of how muscle excitation works. You'll have these big chain reactions of electrical excitation as the voltage starts changing, ion channels will open, which will allow electrical charge to cross the membrane and create even more changes in electrical separation across the membrane. So it'll be fun as we'll watch voltage gated channels create all sorts of cool electrical activity in your neurons and your muscle cells. All right. We will be spending more, if this is seeming a little like what's going on, we're going to spend a lot more time digging deep into voltage gated channels and going more into detail exactly how that works. What other things can gate an ion channel, can control whether it's open or closed?
Some kind of a messenger from another part of the body, like a protein that will attach on the outside of the cell.
Exactly. So the kind of generic word for these signaling molecules is ligand. So I'm gonna, if you don't know this word, it's a word we're gonna use a lot in this class. Ligand gated. So they can be voltage gated, they can be ligand gated. Ligand would be like a neurotransmitter or a hormone or some little molecule, some signaling molecule. So again, ligand is typically some signaling molecule, that usually binds some kind of receptor.
I have a quick question. Speaking of ligand, so can these be dysfunctional? Are these the ones that are kind of dysfunctional with neurotransmitters?
I'm not sure what you expect.
I guess I'm wondering with these gates, if there can be dysfunction with them.
Yeah, absolutely. Yeah.
So what does that mean then? Is that where medication comes in? Is that where, or what would happen if these were dysfunctional?
I mean, there's so many things that can be wrong. There could be some genetic issue. There could be some things caused by some kind of infection that's messing with the signaling that controls the opening or closing of things. So that question is like really broad. So it would be like, kind of like, it's almost like saying sick. Like that means so many different things. So I would say if things go wrong with these systems, it definitely leads to problems, but there's so many ways things can go wrong, so many different kinds of things that can go wrong that, it's not something I don't think I can address in a.
Okay. Yeah, I didn't know if we were going to go over that, like go more in depth with that, or just kind of like base.
We'll be talking about a variety of different things. And this is also a lot of meds work by acting as ligands and mimicking naturally occurring ligands. That's how a lot of drugs work. Like, if you take morphine, it's going to mimic a ligand naturally occurring opioid and goes in and is going to bind receptors and, turn on these pain relieving pathways, or things like that. So,
Okay. Yeah, that's where the medication, that's what I was thinking, where the medication kind of comes in to restore, help, or facilitate.
right. So, but we'll see. There's so many different options. So after we in another week, if you can ask that question again, if there's clarification, but we will be covering, I think, some of what you're asking about right now. So ligand gated, again, these could be, these could be neurotransmitters. They could be hormones. They could just be general molecules. You know, the way that your taste buds work is you have ligand gated channels in your taste buds that recognize sugar. And when they bind sugar, it opens up ion channels that create an electrical signal to your brain that you're tasting sweet. Right? So the ligand might just be kind of environmental molecules that you are detecting and turning on electrical signals that then go to your brain that you become aware of. So we're going to see lots of ligand gated channels. Again, it can be either neurons talking to each other, hormones affecting cells, and like I said, just your cells responding to molecules in their environment and opening up ion channels, creating voltage changes across the membrane, which lead to electrical signals to your nervous system. So ligand gated is going to be important as we continue on. And there's one other super important one. So voltage gated, ligand gated.
Is it mechanically gated?
Yeah, mechanically gated. So this is literally just a deformation of the cell membrane, results in ion channels being open or closed. This is what touches. Like, how do I know that this like Apple Pen is touching me in my face? It's because I have these mechanically gated channels in these little receptor cells. And as the cells are getting stretched and deformed, as my skin gets pulled, ion channels are opening up and sending a message to my brain with these electrical signals that I interpret as touch and pressure. Even the way you're hearing my voice right now is, my voice is vibrating, going into my little microphone, vibrating ultimately the speaker on your computer, which is vibrating your eardrum, and that little tickle on your eardrum moving back and forth is ultimately opening and closing mechanically gated channels in your cochlea that are creating electrical signals that ultimately you perceive as sound. But all sound are mechanically gated channels responding to the vibration and wiggle of your eardrum moving back and forth. It's kind of a weird thing. It's my talking is kind of tickling you on the inside of your ear, and you're interpreting that as sound. But the only reason it works is mechanically gated channels are opening up for ions to cross. As the ions move across into the cell or out of the cell, it creates voltage changes, which are going to become these electrical signals that go into your nervous system. So gated channels are going to be super important as we continue on in the semester. So that's gated channels. Next, so any questions about these ion channels? All right, now we're going to move on to another class. We're going to still continue talking about transport proteins, but a slightly different version. So carriers. Carriers tend to be for kind of polar molecules like glucose and amino acids. The thing that's different about a carrier compared to a channel is that they're not like a tunnel. they're kind of more complicated. I like to think of them more like a revolving door. So for instance, let's say this is a glucose carrier. I might have a glucose that binds on here, but it doesn't just flow through like a tunnel. It has to go here. There's this kind of conformational change, like ka-ching, ka-ching, and then the glucose ends up on the other side. So there is an actual kind of change of shape that has to happen as this thing does its job to move this glucose from one side to the other. The glucose will still move along its concentration gradient. So it's still going to move from high to low concentration, but there is something kind of different going on because there is this, think about if there were like 100 people inside a building and they all want to get out of the building and all they got to use is a revolving door, there's going to be a limited number of people that can get out per second because the revolving door has a maximum number of people that fit in the revolving door and a maximum speed that the revolving door spins around to get the people in and out of the building. So if you have a bunch of glucose trying to get into a cell or getting out of a cell, it's still going to move in or out according to its concentration gradient, but there's going to be like a maximum rate that things can move across using a carrier. So these are rate limited. So in fact, this idea, we call this facilitated diffusion. Facilitated diffusion if crossing membrane using a carrier. So why are we using this other word? Why are we distinguishing it from regular diffusion? Because we have to worry about rate limits. So for instance, if I have, the classic graph of, concentration gradient. Concentration gradient just means the difference between the two sides of the membrane in concentration and like diffusion rate. Right, if there's no different, if I'm looking at, here's my cell, I have a certain amount of stuff inside, certain amount of stuff outside, inside. If there's a balance, the same amount, no concentration gradient, gradient is the difference. If there's no difference, there's not going to be any movement across the cell membrane. The greater the differences between the inside and the outside, the faster things are going to move from one side to the other side. So this is normal diffusion. Normal diffusion, the greater the differences between the two sides, the faster things move from one side to the other. However, if we're doing facilitated diffusion where we're using carriers, it kind of starts like that. But at some point you hit that maximum rate, again, like where the revolving door is working as fast as it can. And there's some like Vmax here. So this is this facilitated diffusion. Whoops. So this is going to be important. It actually comes up in physiologically important contexts to actually understand that if things are crossing the membrane using a carrier, that there is a maximum rate that things can cross. This is going to be at the core of why you end up having sugar in your urine if you have diabetes. Like, normally in your kidney tubules, the nephrons are reabsorbing the sugar back into your body so you don't pee out any sugar. And normally the capacity to reabsorb, keep the sugar in your body is fine for your normal functioning body. But in diabetes, where you have very high levels of sugar in your bloodstream, it overwhelms the ability of the kidney tubules to work properly because they're using carriers to move glucose from one side of a cell membrane into another, and they can't do it fast enough in the disease condition of diabetes. So we're going to be seeing facilitated diffusion actually having physiological consequences as we go on in the semester. It also makes a difference, like if you start taking tons of one kind of amino acid, and there's some carrier that lets across a certain subfamily of amino acids, you might actually limit the ability to absorb other kinds of amino acids by taking too much of a particular kind of amino acid, because there's only a certain amount you can absorb using a particular kind of carrier. So make sure you kind of understand the difference. Facilitated diffusion, it's still passive transport. Things are still moving according to their concentration gradient, but the difference between facilitated diffusion and just normal diffusion is at some point you're going to hit some maximum rate that things can cross the membrane using the carriers. Oh man, it's 9:30. So transport proteins. We are going to continue talking about that. There's also going to be co-transporting, coupled transport, I should say. So these symports and antiports we're going to be looking at in a little bit. There's also going to be kind of active, like pumping, where we actually use energy to push things against their concentration gradient. But we will look at that in a little bit.